Okay, let's learn. I'm going to use up a lot of time today explaining the gene expression project, so that'll be probably nice for you guys. I just got a nice question, and for those of you who will be taking the exam this week, which should be all of you, I'm going to share. Someone asked me, okay, so the cyclins change at different phases of the cell cycle. Is the CDK the same? Is it one CDK that just different cyclins activate at different times, or are they different? And I didn't actually answer the person's question. He answered it himself because all he had to think about was about like CDK is a kinase, and a kinase is going to phosphorylate something. And the only thing really that determines what's phosphorylated by the kinase is whether or not the kinase can interact with the protein, right? If you're a kinase, you're only going to phosphorylate proteins you can bind to, right? You're not going to phosphorylate something that's on the other side of the cell, right? Yeah? Who's like, what is she talking about? If anyone's really confused right now, then we need to have a talk. But you know what I'm saying, right? If you're transferring a phosphate group, you got to transfer it to something close, right? You're going to throw the phosphate group across the cell? No, right? Okay. Anyway, so that kind of answered the question, right? Is it the same CDK or is it different CDKs throughout the different points of the cell cycle? It's going to be different because there are different substrates that are phosphorylated during the different phases of the cell cycle, and the CDK needs to be able to interact with them. So, in case anyone else had that question, it was a good one. Okay, you guys, Monday morning. Hey, I'm tired too. Just letting you know. My son was like, what was I was writing your stupid exam until like 12:30. Yes, I did wait till last minute. And then my son was up at like 1. Why? I don't know. And then my daughter woke up at 2 and stayed up till 4. And I feel like I should, if I'm going to be this sleep deprived, I should have an infant or be out partying, right? Not like have older kids who can sleep through the night. But anyways, I'm going to power through, and I'm glad I'm not taking the exam. Here we go. So here's the business of the week. You have an exam today. Please do not discuss the exams with other sections because I'm doing it different this time. It is the same exam. So if you go and talk about it with someone else, you're really helping, so you're going to help someone else, but you're not helping yourself at all, and they're going to do better than you. They're going to learn from your mistakes. So why would you want to help other people? Right? You want them to do badly. Like, just please don't cheat. That's my request. And it'll be obvious because I'll see it in a marked improvement throughout the week. And then what I'll do is I'll scream at you all. Just call you names. Okay. The exam this time is going to be one short answer that's fairly significant, but there's 3 of them to choose from. So you can, that way you won't die. Okay. Let's talk briefly about the gene expression project. I was going to talk about it last week in recitation, where we could have plenty of time to discuss, but we had a snow day, and then, well, that was that. So we're going to talk about it now, and I really want, obviously the whole class is not here. They're at a disadvantage. I know who these people are who never come. I know who you are. No, I don't have any idea who they are. Who thinks that the professor spends any time thinking about you? Like, when you know people sometimes are like, that professor hates me. No, we don't hate you. We don't know who you are. You're like one of 80 people. Anyways, gene expression project. I want you guys, everyone here is probably going to at least be in a group with someone else who's not here. So pay attention, ask questions, because I want you guys to come to recitation next week with topics picked for the project. All the information is already posted on Blackboard from last week. If you had already looked at it, fantastic. If not, you still have all week to look at it. Okay, so the basics of this project is that you are a researcher working on CMV, and you are starting a biotech company, and you are competing for funding. And whoever has the best idea gets the funding. Okay? So each group is going to work together to form a proposal about what they want to study based on a set of preliminary data that I give you. Okay? So the topic is CMV, cytomegalovirus. It's a virus that we're all infected with it. We're all fine, but it's really a big problem if you're immunocompromised or if you get infected when you're pregnant and your baby gets infected. That's really bad. It's called congenital infection. So it's a problem because there's no significant way to treat congenital infection, and so these babies that are born with this, it's bad news. So I have a collaborator at Hopkins who I do some work with who studies this, and I did this. This is actually real data that I generated along with a collaborator to do some other research, but we're using it for this exercise. So your challenge, investors are looking to fund biotech companies with novel ideas for CMV research. You're competing with other biotech startups for $10 million. Who believes they're really going to get $10 million? Okay, good. You're going to get $10 million in startup funding for 5 years. That's a lot of money, but not really. It's amazing how you can spend money in science, especially when you're paying salaries. And that's where most of this would probably go, right? What are you going to pay yourself as a CEO? Probably at least $1 million a year, right? Okay. Your job is to propose a novel area of CMV research based on preliminary gene expression data. Okay? This requires you to look at the data, to think about the data, to analyze the data, to read the literature, to make sure you're not doing something that's been done before, to get background information. I'm actually going to treat you guys like graduate students, every single one of you. And you might be scared, but you're going to be fine because last year, bless you, everyone did fine. There were a lot of people who started out really confused and in the end loved it because they finally got it and enjoyed it. So go with it. Okay, so individual startups, each group will be judged based on novelty. So if everyone shares their idea, you don't get these 10 points. So you want to hide your information from your competitors. A written research proposal, fairly short, 35%. So you're gonna, and there's information on exactly how to put that together. A brief presentation, very short, and an oral defense. So on the last day of recitation, we're all gonna get together, and everyone's gonna do their brief presentation, and you're gonna have to defend your idea, not only from my questions, but from everyone else in the classes. So we're not going to do this in lecture, you're going to do it in recitation, but don't forget everyone's looking at the same data, so everyone's considered the same projects, and people have decided on one or the other based on what they thought was the best idea. So people are going to know what you're talking about, and they're going to have questions. And a team evaluation, 10% of the grade. So if someone's doing nothing and trying to get a free ride, the rest of the team should let me know because they should lose these 10 points. No one should get an A when they don't do their share. That's always so embarrassing, right? When you're sitting in class and your phone goes off. You know what's even worse is when you are giving the talk and your phone goes off. And it's like over there. And you're like, oh, stop ringing. Okay. So preliminary data. Let's talk about preliminary data. So what we did, we looked at the effect of CMV infection on immune-related gene expression. So we took human foreskin fibroblasts, cells, and infected them with CMV. And we harvested them at time 0, so that's an uninfected control, 2 hours post-infection, 5 hours post-infection, and 24 hours post-infection. And then we isolated all of the RNA, because remember we're looking at gene expression. That means induction of transcription, and then later on translation. And cDNA was synthesized, and that's the template for gene expression analysis using a real-time PCR array. And so there's a link there. You can go to the website and look at the array, look at all the genes, and whatever you want to do. Read more about it. But a real-time PCR array is basically 96 well plate. I think I have a, yeah. So this is from the company. There's 96 well plates, and you buy them, and in each well is a primer pair for a gene in a certain pathway. So this is the antiviral response, the antiviral array. So every gene that the bioinformaticists that work at this company, every gene that they've found identified with that's involved with the immune response is included in this array. Each little well, the 96 well plates, there's something like 70 genes that are being analyzed, and you take your cDNA from each, from time 0, from 2 hours, 5 hours, 24 hours, and put each one on a separate plate, and you can follow expression of these different genes over this time course. And then you put it on, you do real-time PCR, and you get this magic here, and then you analyze it, and then you can understand whether genes are upregulated or downregulated in response to infection. Okay? So this is the data that comes out of the machine, but I've done you guys a favor and analyzed it for you because this is not a class in data analysis. And what I'm giving you all the data, but what you can mostly focus on is the full change or full regulation. So there's actually an Excel spreadsheet that's on Blackboard, and you can look through it and you'll see there are different tabs on the spreadsheet. There's the gene table, which is just a list of all the genes, and it has the accession numbers and the names of the genes and anything you need to know in order to look up the function of the gene. You're going to need this because if you see something that's upregulated, you're going to want to know what the gene is and what it does. There's the average CT. This has to do with when the gene amplifies. So a low CT is low expression, a high CT is high expression. A low CT is high expression, and a high CT is low expression. Then there's the delta CT. This is a way to normalize the data because just because they're taking RNA from different cells doesn't mean that it's equal. And since we're counting copies of RNA, we want to have a way to normalize it. And so we use housekeeping genes. These are genes that are constitutively expressed, like actin, GAPDH. Those are the ones that I like the most. Ribosomal proteins. These are things that are not going to be induced by viral infection, hopefully. If they are, we throw them out. Actually, one of the genes that is included in this array is beta-2 microglobulin, which is part of MHC class 1, and obviously that's going to be affected because it's an immune response-related gene. So we threw that out because it's not useful to normalize your data based on fluctuating data. And then the most important one for you guys is full change, full regulation. So after we normalize the data against these housekeeping genes, then we normalize it against time 0, and you get everything relative to time 0. So if at time 0 it was this number and it went up, it was upregulated. If at time 0 it was this number and it went down, it was downregulated. And up and down regulated is the way to start thinking about things. Okay? Questions so far? I'm kind of blazing through this. I would like a lot of questions because I want to make sure everyone understands it. I'm going to show you a few screenshots of what the data looks like. So this is what the gene table looks like. And it gives you the position. That's just where it was in the 96 well plate. You don't need to pay any attention to that. It doesn't make a difference. These are accession numbers from Unigene and RefSeq. The RefSeq numbers from NCBI. And then this is the actual name of the gene. Very useful, right? Description of the gene. Please don't limit yourself to just reading the description and making your judgment based on that. Look it up by going to NCBI gene and actually put in these numbers or the gene name and read something about it. You don't need to go to, you don't need to start reading primary literature, but when you go to the NCBI gene, you can actually get a little description of what each gene does. Alternative gene names, because sometimes there's a lot of different names for genes. And then some catalog number, which we don't care about. So you have that for all the genes. Okay, so when you start to see things that you're interested in, you are going to be able to go back and see, well, okay, what gene is this? What does it do? And this is what the full regulation tab looks like. So this is already normalized data. So the CT data is not useful to look at. Only really the full regulation or the full change are useful to look at, because the CT is not normalized at all. So if you, like, by mistake harvest twice as many cells in time 0 versus time 2 hours, you're going to have twice as much RNA in time 0, and that's not useful because you're counting how much RNA is there, right? So you need to. That's why you have this normalization step. So don't get stuck looking at the CT data. So you'll get this fold regulation, and this is group 1, group 2, group 3. Those are the different time points, 2, 5, and 24 hours. You might say to me, well, where's time 0? Well, it's normalized against it. This is all relative to time 0. So for example, this gene here, ApoBec3G, is upregulated 2.5-fold at 2 hours, 5 hours, and then it goes up a little bit more by 24 hours. Okay, that's how you would look at it. This gene, CCL3, is downregulated at 2 hours, and then nothing really. Now the thing to keep in mind is upregulation or downregulation is only significant if it's above 2. So if you start to see something that's 1.5-fold upregulated, that's kind of within within the margin of error. So you don't want to pay attention to that. Yeah. So blue and red. Look at blue and red. Yes.
What are comments? Are they significant?
Comments you don't need to pay attention. You just go with the data. Comments have to do with sometimes when things are. So this was done by a program, so it spits out a bunch of comments based on if you start with very low level and you go very high, because they think that might be an error. But when you're looking at the immune response, things often are low to really high. So don't worry too much about that. And if you were studying this in real life, what you would do is you would validate these. You would say, oh look, ApoBec3G is higher. That's really interesting. Well, let me repeat this experiment just looking at ApoBec3G. Let me look at protein. You look at a few different things to make sure it's real. In this case, you're trying to figure out what are you going to look at. And you're going to look through all this data and say, oh, look, like all these interferons are up. Or, oh, this virus downregulates it. This is really interesting. Okay, so that's what the data looks like. I want you guys to spend some time looking at it and familiarizing yourself with it and trying to figure out what you're actually doing. Okay, so what do I do with this data? What are you talking about? I definitely had a lot of people at the end of the project last time I did this say to me, I was so lost until the end. I felt like I had no idea what was going on until like the last minute. And that was unfortunate, but I guess sometimes you have to go through that to get to understanding. So if you're lost, come to me fast, quickly, so we can get you unlost. But I think the reason I give you this project is because the whole point of gene expression is that you're expressing genes in response to something, and I say it over and over again, but I feel like this really shows how it happens, right? You have a virus infecting cells, genes are going to be expressed, or actually, sometimes genes will be turned off. That's very interesting too, because viruses like to shut things off that they don't want around. So, what do you do with this data? Look at it, right? Look at genes that are up or down regulated. Determine if there's a trend over time, right? If there's no significant trend at all, is it interesting to you? Maybe there was a mistake somewhere, I don't know. But if it's, let's say, it's something that's at 2 hours and 5 hours and goes away at 24 hours, think about the virus. What might the virus be doing? So this might require you to think a little bit about a virus. Not too much. You don't need to know virology in order to do this. You might just need to think, okay, well, at 2 hours it just infected the cell. 24 hours, something else is happening in the life cycle. Consider the function of the genes and whether groups of genes might work together in a pathway, right? Some genes are going to work together, and you might want to see, oh, well, these are all downregulated together. Oh, that's really interesting. Oh, well, this one's downregulated, and it works with these other genes that aren't affected. Does that mean anything? Those are the really interesting stories. Consider why the virus might want, I know viruses don't want to do things, why the virus might want to do this to the cell, right? Because don't forget, one part of it is the cell responding to the virus. The other part of it is the virus dealing with the cell. Viruses have ways of shutting things off. So that's very interesting. So be prepared in recitation next week to give me a topic for your proposal. That means find a time to meet with your group this week and divvy up who's going to look at what. It's something like 70 genes. You don't need to look at them all. You only need to look at the ones that are significantly up or down regulated. You could completely ignore the ones that are not changing, because why would you want to focus on a gene that doesn't change, right? So divvy them up, and each of you guys take a certain number of them and dive into the information. Dive into whether or not anyone's done work on this, and what the genes do, and what people have found so far. And be prepared to form a story in your head. This gene seems to be turned on. Oh, other people found it was turned on, and it was involved with this. Oh, well, this is involved with that pathway. Oh, look, that gene's also in this. So you have to go back and forth a lot between the literature, functions of genes, and just your brain, just using your brain to think about things. A good topic, what's a good topic? It's something that's interesting. It's logical. It's novel, so it hasn't been done before, and it's significant. Further researching this is useful because it will help people, or I don't know, you'll cure all babies of disease. I think, yeah. Okay, so that was a whole lot, but you guys are going to be okay. So does anyone have questions about this? Yes?
When you say a topic, where is it that you're looking
I'm looking for, you pick a gene or a group of genes that are, a gene that's either up or down regulated and it's interesting to you, or a group of genes that appear to be up or down regulated together, that work together, that's interesting to you for some reason. Yeah. Yes?
So is that group a recitation group?
Your group is your recitation group. Yeah, so hopefully by now you all know each other well and have figured out, who's good at what, and as someone, there's always something someone can do. And sometimes just sitting together and talking through your ideas is really useful. I know people don't have a lot of time, but I highly encourage Google Hangout, okay? If you can't find a time to meet on campus, people always use that as an excuse. I have 3 kids, I still find time to get to meetings, so I don't hear it, all y'all. Google Hangout, you can do it 10 o'clock at night while you're in your jammies, okay? If you don't understand Google Hangout, come find me. I'm sure someone in each group maybe has heard of it, right? Who's heard of Google Hangout? Okay, there's a number of hands. Okay, that's where you can get I don't know what the limit is, but I've easily had about 5 or 7 people at a meeting on Google Hangout before. So if you have a computer, you can do it. Okay, any other questions? Easy as pie? Is pie easy? Who thinks making pie is easy? Crust is so easy if you use a food processor. I'm just saying. Food processor crust. Okay. Okay, so is everyone, are you scared? Is that why I see the dead faces? Are you happy? Who's excited? This is never due. It's going to go on forever. No, so if you look at the syllabus, it's a multi-week project. So next week you come, thank you for that question. Next week you come with your topic, and then I'm going to have to check it, but I believe this goes until the end of the semester. And so every week, we're going to spend part of recitation with me going around and talking to you guys about your ideas, so you're not working in the dark. You're going to bring your ideas. I'm going to say, well, why is it interesting? What have you found that's useful? Give me your logic. And we're going to go back and forth. And if you can't defend your idea to me, then I will let you know that right away, so you can ditch your topic and change it. And then the rest of that recitation will be doing a paper like normal. Okay. Yes. Okay. Any other questions? Who loves CMV? Okay, we'll ask that question again at the end of the semester. Okay. So let's, as we begin our unit on gene expression, let's just start with a brief discussion. Does everyone need to switch gears? Switch gears. Okay. What is gene expression? I forgot my list, so I'm going to have to call on people and point at people and hopefully remember names. What is gene expression? Yes.
When proteins are made using the DNA template.
Proteins?
Well, RNA, which is then made into proteins.
Okay, so when RNA is made using a DNA template and is then translated to proteins. Okay. Give me more. Yes.
It's in response to a signal.
So it's happening in response to some kind of signal somewhere. Like, for example, infection with CMV. Yeah. Okay. What else? Who thinks that the transcription part, which would be making the, I should have said that, which would be making the RNA is gene expression, or whether it's making RNA and making protein, or if it's just making protein? Who has a feeling about this? I've actually gotten into arguments with people about this. What does gene expression mean to you? Is it the transcription part, or the translation part, or both? Full response? Yes. Making a functional protein that actually does something. So I challenge that one. I challenge it. Can you express a tRNA? Is there a gene that encodes tRNA? Okay, what is tRNA? Transfer RNA that's involved in translation. Yep, well, so it never becomes a protein. Okay, does anyone want to add to that? Yes.
I would say it's probably everything since it's about just changing the way that the cells actually react on a basis.
So it's about changing everything. So you mean it's about RNA and protein.
So probably getting changes.
So it's about affecting how the cells acted in response to something. Okay. Who likes this conversation and who wants to go home? Okay, fine. So when does gene expression happen? What turns on gene expression?
I guess a signal outside the cell.
There's some kind of signal coming from the cytosol. Yeah. And where's that signal coming from? Where? The environment, okay. So it could be coming from outside the cell. That could be one place it could be coming from. So gene expression happens in response to something, right? There's one thing you leave this class knowing, it's that. So how does a cell know to express a gene? Yeah. Yeah. There's some kind of signal saying you need to upregulate the gene. Okay. So let's go with, and so there's some signal in the cytosol, and how does it get though? How does it actually get to gene expression? So there's a signal in the cytosol. Where does it need to go to affect gene expression? To the nucleus. Something needs to go into the nucleus. And now, with gene expression, are all genes expressed, or specific genes expressed, or how does this work?
Specific.
Thank you. Specific genes in response to something. Okay. Good. Good. Someone's listening. Okay. So, good. So why would a cell want to express a particular gene? Like, why would a cell want to upregulate Hemoglobin, yes.
Well, the cell survival, like it's gonna express whatever it needs to survive.
Yep, yep, it's gonna express whatever it needs to in order to survive. So let's say you have a situation where there's low oxygen, okay? Then you're gonna get upregulation of a whole bunch of genes that are gonna allow the cell to adapt to low oxygen, such as genes that, like hemoglobin, right? Or genes that, that create new blood vessels that are involved in angiogenesis, right? Because that helps to deliver oxygen. So in the middle of tumors where it's really high toxic, there's no oxygen. Usually you have high expression of these genes that are trying to create new blood vessels. Growth factors. Okay. I just want to go into this, making sure everyone has a good way of thinking about gene expression on a very broad basis. So the reason I put this picture is because you've got your whole cell, and then there's something outside binding to some kind of receptor, and then these triangles going into the nucleus. I guess those triangles are signaling proteins that are sending the signal in the form of some kind of transcription factor into the nucleus, and it's going to bind to some kind of somewhere on the DNA, right? And that's going to determine what gene is being expressed. So transcription of a certain RNA, which will then most likely be translated to protein, but sometimes you get transcription of things that will never be made into protein, right? Who's ever heard of RNAs that don't become protein? Okay, good. Shout them out. Give me an RNA other than the one I just told you that doesn't ever become protein. MicroRNAs, right? MicroRNAs are involved with destabilizing other RNAs. What else? There are a lot of RNA subunits to lots of things, like we just learned about telomerase, right? Ribosomes, yeah, okay. And then obviously tRNA, which I said before. There's a lot of RNAs. There's RNAs involved with splicing. So there's a whole lot of RNAs that never become proteins. Okay, let's move on. Transcription. Okay, so transcription is the first step in the process of gene expression. I was looking at this and I decided I actually didn't like this wording. I was looking at this morning and I was like, I feel like this could be rewritten. The title of this slide can be rewritten to be more accurate. Is transcription the first step in the process of gene expression? Not really, right? That's kind of like a midpoint in a weird way. Right? What do you think the first step might be in the process of gene expression? A change in the environment, which will trigger signaling of some sort and some kind of protein, a transcription factor coming into the nucleus. That's really the first step, right, in the process of gene expression. But I guess if we're talking about the technical term of gene expression being transcription, being, I don't know, so then I guess transcription. Anyways, so let's think about this. You've got your DNA, which has all your information, and a copy of it is made into RNA, and then it's translated into protein. You guys have seen that a million times. I just put this in here because, yes?
How does the cell know which strand to use to make mRNA?
How does the cell know which strand to use to make mRNA? I think it depends on, because I think you can have on both strands. Genes are on both strands. And it depends on where transcription factors bind and how RNA polymerase is recruited. Yeah. Good question. So I put this in because all the time people say to me, not all the time, it was one very, very scary time, where someone said to me, wait a minute, you need to copy all of your DNA before you make your RNA. And I really want you guys to not carry that out of this classroom. To remove that from your brain. You're not copying, you're not doing replication, transcription, translation. You're never ever doing that. You're doing transcription of the gene that you want to express and then translating that mRNA. Okay? Does anyone wonder what I'm talking about? Good. Okay. Okay, so this is transcription in a nutshell. We'll go into it in more detail on Wednesday. So you have a promoter, and then you have a terminator. So that's where transcription is going to start, and that's where it's going to end. And the whole point of transcription is getting RNA polymerase to the promoter. And there's different ways that happens, but it mostly has to do with other proteins that are going to bind and recruit RNA polymerase. Okay, so first the promoter needs to be recognized in some way, usually through binding of other proteins, that then recruit other proteins, that then recruit RNA polymerase. Once RNA polymerase binds the promoter, it opens it up, and then you got a transcription bubble, which is similar to the replication bubble. Except for you're not doing bidirectional, you don't have these bidirectional replication ports, you're just making one copy. You have a template strand, and you make one copy, and then you just copy the whole thing. And then, unlike with replication, where you have base pairing of that new DNA with the parental strand, you don't have that at all. This bubble kind of moves along and closes behind it, and the RNA comes out of RNA polymerase elsewhere. Yes?
What is a nutshell?
Did I say nutshell?
On the top.
Oh, in a nut. That's like very basic. Yeah, there's no nutshells involved. No, what I mean by using that is just like this is a very basic version of transcription. No details at all here. Who thinks this is very detailed? I call this the no detail version. Remember the In a Nutshell books? No, I think they went out of business. I have a bunch of friends who went into publishing because I have a former life where I would have met a lot of people who do that. And I went to film school for a little while, and I also had a lot of writing and publishing people. So I know all these people in publishing. So a friend of mine worked on these, there's like the For Dummies books. The competitor of the For Dummies books were in the In a Nutshell books. So it's like the simple, home buying for dummies. I bought that one, by the way. Home buying for dummies. And so the In a Nutshell books were more for programming. So it's like Unix in a nutshell. Perl in a nutshell. Yes. Do you know the In a Nutshell books? No.
If you have a gene that's constitutively expressed, would the transcription factors always be bound or they're associated from the gene?
If you have a gene that is constitutively expressed, would transcription factors always be bound, or would they ever dissociate? Well, really, it's all about RNA polymerase being bound. And a lot of transcription factors leave once RNA polymerase is there. My guess is that's a good question. Well, you know what, that's a really good question. A really good question. So who wants to try to answer that question? If you have a gene that's constitutively expressed, would you always have transcription factors bound to the DNA, or would they ever dissociate? Are you raising your hand because you have an answer?
I think it's not that they're constantly being expressed, it's that they're being expressed to the same rate, all the cells. So the transcription factors are going to be common between the cells that cause them to be expressed at the same level.
Well, what if you have a gene that needs to be constantly expressed? You have a protein that you need to constantly make. So
Well, you make as much as you need and then you stop making it and make more.
Okay, so the discussion that's happening over here is really about if you need a protein at a high level in the cell all the time, how do you maintain that? That's the real question. And there's multiple things that contribute to the life of protein. One is rate of transcription. One is stability of RNA. Don't forget, once you make that RNA, you don't translate it once and it goes away. It can be translated over and over and over and over. So if you've got highly stable RNA, then you're going to have it present for translation. Then the half-life of a protein. Proteins, that need to be used all the time are probably going to have a higher half-life, so be more stable than proteins that need to be used for a very short period of time. So it's a big question with a lot of parts to it. But I think that was a nice question because it kind of makes you have to step back and think like, well, what factors affect whether a protein's going to be available for express or a protein's going to be around in the cell all the time, right? Did everyone follow that a little bit? Are you satisfied? Okay. Sometimes I feel like it's like my oral exam all over again, every day. When you get your PhD, you have multiple oral exams usually, where they just question you for like a million years. Sometimes I just feel that way. That was a good one. Okay. Any other questions? Okay. Oh wait, did I finish? No. So then you get elongation and then termination, and then you've got your RNA, and it hangs out until it's translated, and then translated, and then eventually it's degraded, often through removal of poly-A tail or fibrin cap, or through the activity of microRNAs that like to target specific RNAs for degradation. Okay. I think this is the last slide for today. I'll leave you with this thought. So RNA polymerase, the core enzyme, is conserved among bacteria, archaea, and eukaryotes. So that must mean it's fairly important, right? And so this is what it looks like. So we're going to go, I'll let you guys go a few minutes early so you can get food before the exam if anyone needs food. And we'll learn a lot more about RNA polymerase and transcription on Wednesday.